Hi all, welcome back to Microbiology Lectures. Today I'm going to talk about cytoplasmic membrane and how they facilitate energy transfer. Also going to talk a little bit about the movement of molecules across the cytoplasmic membrane. Okay, we talked more last time about osmosis and diffusion. We're going to go into a little more detail about how things move across, but also what the potential kind of roadblocks or ways at which bacteria control what moves across. Okay, so the first thing I'm going to talk about is, again, remember that bacteria and archaea are these little packages of life, and everything that occurs within, or to keep that thing alive, has to occur within that membrane. And that includes the ability to break down carbohydrates, proteins, potentially nucleic acids, but lipids, into energy sources. Okay, very much like we do in our cells through our electron transport chain in our mitochondria. They don't have mitochondria, but they have a very similar system that's embedded in their cytoplasmic membrane. Okay? And so the system is similar, and we'll talk about the similarities between eukaryotic and prokaryotic ETCs, but nonetheless, it's very similar. So first of all, just like in eukaryotic cells, you have to have something that can energize a protein to pump protons, in this case hydrogen ions, across the gradient. Okay, so you might have water being split down here, you might have carbohydrates being split down here, and you're getting these free protons, these hydrogen ions, that are building up on the inside of the bacteria or archaea. And in order to utilize the energy from those protons, bacteria and archaea can generate a proton force. Okay? So to do that, you have to get a bunch of protons outside of your membrane, and they want to leak back in, because remember, we're talking about diffusion, and diffusion is this process of things traveling from a high concentration to a low concentration. So it takes energy to pump it across, because you're pumping it from low to high. Eventually you'd be pumping it low to high, and you build up this gradient over here, and then they need to flow back into the cell. Okay, we often call that a proton gradient or a proton motive force, because it's driven, the energy to generate things like ATP is driven by protons. Okay, and so this diagram's really just showing these proton ports, or in eukaryotic cells we normally call them proton pumps or hydrogen ion pumps, and you can think of that as an electrons moving through the membrane. It's allowing, or it facilitates, protons, these gates in these proteins, to be open and forcing protons across the membrane. Okay, after you get this proton gradient on one side, okay, not shown in this diagram, would be another protein that's embedded in the membrane. Bacteria and archaea, it's embedded in the cell membrane, that cytoplasmic membrane, and that protein's ATP synthase. Okay, similar to our ATP synthase, I'll show you in a second. A little bit of differences, but overall same function. Okay, going to use protons, the energy from protons, to stick together ADP, or adenosine diphosphate, and inorganic phosphate, stick it together to form adenosine triphosphate, or ATP, the currency of pretty much all life on this planet. Okay, so that proton force, or that proton motive force, is really driving the generation of ATP. Okay? What researchers recently have figured out, or relatively recent, is the differences between ATP synthase of bacteria and ATP synthase of, say, eukarya, or organisms that have at least a mitochondrial ATP synthase. Okay? And there is differences. Okay? So you can see that there's different amino acid chains that are building these proteins together. And what really is kind of a recent advancement in antibiotics is to potentially target the differences in these ATP synthase structures. So if you target, maybe the head here, or this motive force up here of ATP synthase, because it's different between the mitochondria structure and the prokaryotic structure, then it's potential that an antibiotic could come in here, interact with this, and cause this to malfunction, but not interact with eukaryotic ATP synthase. And so it would cause the bacteria to malfunction, not be able to generate energy, ultimately die, but eukarya would be fine. Okay? And so these are just advancements, and we'll talk more about advancements in antibiotic medicine as we progress in the course. So, transport of small molecules across cytoplasmic membrane. Okay, we kind of have already talked about this, but if it's not able to flow across natively or naturally, like if it's not a gas or water, then sometimes there is a specific protein for that molecule. Alright, so you might have like a protein embedded in the membrane, similar to like GLUT4, or like a glucose emitting protein, which would allow for a glucose molecule to bind to it and then be pushed in, but nothing else could bind up to that protein. Okay, and there's lots of these. As we learn more and more about the genetics of microorganisms, different microorganisms, depending on what they're using for an energy source. Some use sulfur, some use glucose, some use like all kinds of different things. A lot of these molecules that might be traveling in on a regular basis, they often will have an active site that's specific to that molecule, and then that would allow that molecule to go in, and no other molecules can fit that active site. So when we start finding out all the genetic material of a given species, we can start looking for specific proteins that are coded for that would have that kind of action. One kind of interesting, I guess, component of prokaryotic cells is that, remember, so it's all in one package, and so these organisms have to get rid of waste, and they get rid of waste using efflux pumps, right? And what's kind of crazy is relatively recent discoveries, is a lot of antibiotic resistance is due to these efflux pumps. And what's happening is these organisms, when you're dousing them with antibiotics, the antibiotics is getting through the membrane and into the cytoplasm, but then the bacteria is recognizing that as a waste product, or not self-made, or not something that it can utilize, and it takes that antibiotic and that antibiotic chemical and it forces it back out. So it treats it as waste or trash. And so, important to us, we have to find a way to shut off the efflux pumps of many of the bacteria, the disease-causing bacteria that we're trying to treat with antibiotics. Okay, and we've done so, like we've created inhibitors, because often there will be, remember, these are just proteins are just amino acid chains, and often there will be sites on that amino acid chain at which other molecules can bind to, either alloxeric sites, active sites, or sometimes it's just a matter of you pick the right inhibitor to change the shape of the protein so that protein no longer allows for the pumping of material out. Okay, and one of those inhibitors that we've generated somewhat recently is BU005, and that seems to inhibit some efflux pumps. And so when the antibiotic gets in, if that efflux pump is active, sometimes it's pumped out. If it's not active or it is inhibited, then that antibiotic would sit in here and do what antibiotics do. And it depends on the antibiotic, but some of them stop the genetic material from being able to be replicated. New ones are trying to target, again, the ability for ATP synthase to work. There's lots of ways at which antibiotics can function, and that would be geared towards that kind of efflux pump to not allow for those antibiotics to be pumped out. Okay, so in general, when we talk about movement across the membrane, we can talk about a few different ways. You can have facultative diffusion or facilitated diffusion, where the substance is moving through a specific protein, okay, like an aquaporin or a specific protein that is geared towards that substance. Often this is based on a concentration gradient, only moving through on a concentration gradient, but that this is showing a one-way direction, but often it's both directions, so it has easy access flowing back and forth through the membrane. Okay, you have active transport, which most of you probably have heard of before. Active means that it's utilizing some energy source. There's kind of a couple ways at which active transport works. One, you can utilize ATP, so the phosphate group, that third phosphate group of ATP, is used to energize the protein, which would allow for that substance to flow through. It opens the gate, changes the protein's conformation so material can flow through. Or you can do it like the electron transport chain and you utilize electrons to charge that and pull that material through or force that material through. Okay, and then the third one is what we call group translocation. So this can happen in a couple different ways. So one way is that when a molecule comes through, maybe it binds to another molecule, or it has to be transported by using another molecule, or it's possibly that it's part of a biochemical pathway where you have multiple proteins embedded in the membrane, and one product from one protein is the substrate for the next, or fits in that active site on the next and opens that channel for the next protein. So there's lots of ways at which this can occur, but again, a lot of these processes are very similar in eukaryotic cells, and most of you probably learned about that in a eukaryotic cell. So cell walls of prokaryotes, just to kind of cover this again, strong cell walls, super important for the cell membrane not rupturing, not bursting when it's in solution that's rushing into the cell membrane, so it provides that rigidity. Okay, there's two distinguishing types. We talked about this before. There's gram-positive and gram-negative. Gram-positive typically have a very thick peptidoglycan layer, okay, and that's what's dyeing purple with crystal violet when you're using a gram stain. Gram-negative have peptidoglycan, but it's much smaller layer. It's like a single or 2 layers thick kind of layer, but then on top of that, or on the outside of that, it will have a capsule or a covering. Okay, and we'll cover this here in a second. So here you can see the gram-positive. This diagram's just showing you multiple layers of peptoglycan. They're going to be linked together with cross linkage, and provides a rigid cell wall. But that cell wall goes all the way out to the outside of the organism. Okay, so it's interacting with the outside environment of that organism. The opposite, or the difference here with gram-negative, is that peptoglycan layer is embedded in a plasmic space, right? And you have this kind of outer membrane, which we call a capsule, on the outside. So it's not the first line of defense, or the first line of interaction with the external environment. That capsule is, and that capsule is made of lipopolysaccharides, so fat and sugars together, and that can generate issues with inside organisms, especially humans, and we'll get to this here in a second. Okay, so that peptoglycan layer, it's alternate chains of NAM and NAG, so acetylmyrmic acid or acetylglucosane. And so why is that important? Well, the NAM will have a peptide or a tetrapeptide chain that comes off of it, and so a series of amino acids, and then those chains that come off of those branches that come off will have a peptide interbridge between those two chains. And you might say, who cares? What does this matter? Well, that interbridge, that peptide interbridge, is how penicillin works. So penicillin breaks that peptide interbridge, and so it doesn't allow that bridge between these two to form. And so instead of a scaffolding of multiple glycan chains, okay, like you can see here, these are multiple glycan chains. Instead of them being linked together, the penicillin will come in here and cut these and not allow them to form, I guess you could say. So it's like independent chains, but water and other things can flow through. So it's like drilling a bunch of holes through the peptoglycan layer, and it doesn't allow for the protection that it would if it was whole and combined together. And so that, at least in gram-positive, that's how it is. Now in gram-negative, you have similar things. You got a NAG-NAM and the tetrapeptide chain that's coming off of it, but there's no interbridge linkage between them. Okay? So it's a direct link between the tetrapeptide chains, no interbridge. Okay? So you can only imagine if things like penicillin are acting upon that interbridge, then it probably works much better on gram-positive cells than on gram-negative. And if you're thinking that, you're exactly right. So let's look at these gram-positive cell walls. Again, you have that interbridge linkage between NAMs on different chains. Here you have teichoic acid that comes up. This provides an extra layer of protection for the organism. And sometimes you'll have organisms that can kind of communicate using these teichoic acid chains that are moving through the peptoglycan layer. And we talked about it again, like there's a plasma layer between the peptoglycan layer and the cytoplasmic membrane. This just provides an extra layer of protection. It's kind of like a jelly-like form, so things just doesn't flow through, but it also provides like a gap between the peptoglycan layer and the phospholipid layer, or the cytoplasmic membrane layer. Okay. So I mean, in general, often we call it teichoic acid. We just call them TAs, but they have different purposes. Sometimes it might be for communication, cell recognition. Sometimes it's a chemical cue or a protective layer that gives the outside of the bacteria kind of a nasty taste. So if something is going to phagocytize it, it'll interact with the membrane of whatever that is, and sometimes they're not able to consume the material. There's lots of ways in which this works. Gram-negative cell wall, so thin peptoglycan layer, again, okay, not very thick. But to increase the rigidity and increase the structure, they'll have lipoproteins, so fat proteins that are embedded, interact with the peptoglycan, but also are embedded in this other membrane. This other membrane is a lipopolysaccharide, or an LPS membrane, and often will have, a antigen component to it, and this is really what causes toxicity in humans. So when we have a large buildup of gram-negative bacteria in our blood system, even if our immune system is breaking these guys up and destroying them, you still have these lipid antigen structures that are often floating around in your blood, and those endotoxins can really become deadly in some cases, and this is when we see adverse effects to a bacterial infection, and after the bacterial infection has progressed long enough that enough LPS is building up in the organism's body that it will first cause a pyrogen effect, so it'll cause a fever, but then after that it can disrupt organ function and things like that. And so gram-negative bacteria, often we're more concerned with gram-negative bacteria because of this capsule layer that forms outside the peptoglycan, that protective capsule, which is made of an antigen, polysaccharide, and lipid A. And again, it's fat-based, so it doesn't dissolve very well in water. It's hard for our body to get rid of it, and so it accumulates around organs and can travel in the blood system, but it doesn't mix with the blood system. So that can be kind of difficult. The toxicity can build for sure in some systems, some organisms. Okay. All right. Like I said before, there are certain types of antibiotics that can target peptoglycan. And so, like the first antibiotic we ever discovered, penicillin, it interferes with that linkage between peptoglycan synthesis, or between the peptoglycan chains. So it prevents that cross linkage. So if you have that penicillin molecule come in, it doesn't allow the cross linkage to happen, which means the cell wall becomes porous, and so it allows for openings, and it doesn't link them completely together, which eventually causes the organism to be lysed and break up, because water's still flowing in, and now they can't control, or they have no rigidity to themselves, and so they, in changing environments, they tend to kind of rupture and blow up. So again, gram-negative bacteria are kind of not usually affected as much by, say, penicillin, because they just have less chains of peptoglycan, but also they have a capsule that covers it. So the penicillin has to penetrate through the capsule, which can be difficult, and then get into the peptoglycan. But even if the linkage is broken in the peptoglycan layer, they still have that capsule as extra protection. So normally we see that gram-negatives are typically less affected by that or by that penicillin, or any antibiotic that would affect the peptoglycan layer. Okay? And naturally we can do this, and we have lots of systems naturally. Lots of organisms on the planet have natural systems that can fight this off. One enzyme called lysozymes or lysosomes. These enzymes they work in the same function, work with the same premise as penicillin, and they don't allow for that peptide bond to form, and the cross linkage doesn't form, and so the cell wall is porous. So, and we find this enzyme a lot in places where there's a lot of direct contact with bacteria. So, like your eyes, there's a lot of fluid there. The layers of cells around your eyes are very thin, and you can get lots of bacteria in that region. Saliva, same thing, lots of bacteria in the region. We need thin layers in both those regions to transmit signals, versus things like your skin, which is a lot thicker, even though it comes in contact with massive amounts of bacteria. Typically it doesn't need an enzyme to break it down because the skin layer is thick enough that it protects it. So you see these enzymes in tears, saliva, other body fluids, and again, works the same premise. And like penicillin, it's also more effective against gram-positive bacteria. All right, I want to kind of wrap it up with a group of bacteria. I know when we first introduced bacteria and archaea, I said that they all have cell walls, but biology, because everything's always evolving and there are almost no laws in biology because nothing holds true at all times, there are bacteria without cell walls. The Mycoplasma species are one of these groups that have no cell walls, so it's just the cytoplasmic membrane. So it's not rigid, but they increase the rigidity by embedding sterols or things like cholesterol inside the membrane. Just like us, when our membranes become extremely rigid, it's typically based on a poor diet or lots of cholesterol being embedded in our membranes. But they utilize that to their advantage because they have no peptoglycan layer. The other thing that happens is, like Mycoplasma pneumonia, humans can get Mycoplasma pneumonia, and we can't really treat it with at least penicillin or lysosomes. So there's no natural effect against the organism because there's no peptoglycan layer. So neither one really works. And we see this happen, to affect young adults fairly often. They'll get kind of a atypical pneumonia, a different type of pneumonia that might present itself as pneumonia, might be treated with penicillin-like structures, but never really, they don't recover from it as fast as you would expect using a antibiotic, and it's because it's a mycoplasma bacteria, not a bacteria with a peptoglycan layer. And then archaea, again, the group archaea, as far as the kingdom goes, they're fairly newly discovered. I don't mean new, like we've known about archaea for a long time, but until modern genetics, until our ability to actually tell the difference genetically between archaea and bacteria, often we were classifying lots of archaea as bacteria just because of where they existed. And then once we were able to genetically differentiate between the two groups, now we're finding that archaea occur in lots of places, like the human intestines, like all over the place where we didn't think they were before. At first we were like, all right, these are just extremotherms or extremophils, and they only occur in extreme environments, the heat vents, the salt flats, these kind of things. But now we're finding out that They occur in a lot of different regions, and they're very, very hard to distinguish between each other, like with a microscope, because the cell structure, the cell walls are very similar to each other. So we don't know. We know that there's a lot of varieties of archaea that have, like, are potential out there. We know that they do not have peptoglycan in their cell walls, and we know that their cell membranes, often the fatty acid tails, are linked together to provide them kind of a different structure for extreme environments. Also, remember when we originally introduced kind of that cytoplasmic membrane, their neck, or the connection between the phosphate and the fatty acid tail, that glycerol connection is different in archaea. But again, with more time, there will be more information discovered. There will be, we'll have better names for all the different varieties of the cell walls that we see. And right now we're just discovering new pieces, like pseudopeptoglycan, which pseudomirin and pseudopeptoglycan are like, the same term, or have the same meaning. It's not quite peptoglycan, but it has a similar structure as like a glycan layer, stringed together outside of the cytoplasmic membrane. And then there's these S layers, and these S layers are often glycoproteins, or some type of protein layer that goes on the outside. And there's lots of different reasons for this. Protection from high salt concentrations, lots of things that are going on. And like I said before, when more information is available on all the different archaeas, we'll have a better idea of what these cell walls are made of and what's the purpose of the S layer. We do know that it can recombine, it can self-assemble, it can be damaged and then fixed. So there is some information that we know about it. Alright, so next time we're going to talk about capsules and slime layers, which often will just add to the defenses of these microorganisms.